Hello, everyone. My name is Avelino Álvarez Ordóñez. I am an associate professor at the University of León in Spain, and I am here today to talk to you about a validation of a protocol that we have carried out within a European project, the European Project Master, to map microbiomes in food processing environments and in foods. So let's start first talking in general about Master, about the European Project Master. Master takes a global approach to the development of microbiome products, foods, feeds, services, or processes with high commercial potential. This is a collaborative project coordinated by Dr. Paul Cotter at Chagas in Ireland. And the vision of Master is to benefit society through improving quantity, quality, and safety of food across multiple food chains, including marine, plant, soil, rumen, meat, brewing, fruit, vegetable waste, and fermented foods. So a wide variety of food production and food chains. We are in total 29 partners from 14 different countries in Europe, well distributed all across Europe, as you can see here in the map. And we have achieved in the last 4 years a wide range of innovations, very interesting results in many different aspects, from biofertilizing strains, biocontrol agents for improving safety in different food chains, probiotics, prebiotics strategies for improving nutrition in animals and also in humans, different animal nutrition strategies to control the methane production in ruminants, and many other innovations that you can see here in this slide. But today I want to talk to you specifically about some innovations related to the standardization of protocols for the microbiome mapping in food industries. This innovation is the result of the work carried out in the Work Package 4 of Master, which is a work package related to microbiomes in food processing and also to the valorization of waste streams and to the human gut health. This work package is coordinated by the University College Cork and also the University Federico II in Napoli, in Italy. And these are the main objectives of the work package 4. So we are our aim were to generate standard operative procedures and tools to map the microbiome of food processing environments. That is what I am going to present you today. But also there are another important objectives in Work Package 4, more related to biopreservation, to valorization of food waste and management of food waste, and also to human gut microbiota and to the development of nutrition solutions for improving the gut health. Relating to the microbiome mapping workflows and tools in the food industry, so I would like to start doing some considerations of why this is something needed. So we know that microbes can colonize the surfaces of equipment and the tools, and that in the industry, and that can be transferred from them to the food product or to intermediates of production during the handling, manufacture, processing, and storage of the foods. Both food contact surfaces that often represent a good niche for microorganisms to persist or even to proliferate, but also surfaces that are not in direct contact with foods, but that they are a potential reservoir of microbes, which over a longer term can be also important sources of contamination. Of course, the industry daily applies cleaning and disinfection procedures, but these are not always effective in eliminating the resident microbial communities specific to each food plant. And the microbiome resident in these processing environments, even after cleaning and disinfection, can have a very important impact on the food quality and on the food safety. Also, those microbial communities, that microbiome in the food processing environments, can significantly change over time in response to different factors, such as the presence of organic residues coming from food, variations in the cleaning and disinfection practices, temperature shifts, etc., etc. So this is why it's really important to have good tools to map the microbial communities colonizing food processing environments. And the truth is that before Master, there were no real protocols and workflows specifically tailored to do microbiome mapping in this type of environments. So our main aim in the task 4.1 of master was to develop rapid food and environmental microbiome testing procedures for food processors to enhance food quality and safety, reduce withholding periods, and reduce food waste as a waste management strategy. So our approach was quite holistic in the sense that we wanted to provide a complete microbiome analysis approach, starting from sampling to the manipulation of samples, the DNA extraction, and all the sequencing steps and computational steps of the bioinformatic analysis of microbiomes. For that, we wanted to create standard operation procedures to be directly applied by the food industry and to validate them in a large set of companies across Europe. So these validation activities have been already completed a while back in 5 different countries of the European Union: Spain, Italy, Ireland, Austria, and Iceland. And these validation activities involve more than 100 food processing companies from different sectors: cheese processing companies, companies producing cured meats, fermented sausages, raw meat, and fish, minimally processed vegetables, etc., etc. And in total, in these validation activities, we have analyzed with our protocols and with our workflow more than 2,000 samples, mainly from processing environments, but also from raw materials and products, and even from operators. And these are the partners that were involved in all the validation activities. You can see partners from Spain, Italy, Ireland, Austria, Iceland, and Italy. So these are the main steps of the mapping protocol, and we will be start talking individually about each of these steps. So we will be starting by the sampling in the food facilities, then the sample manipulation, the DNA extraction, the sequencing, and the bioinformatic analysis. So let's start with the sampling in the food facilities. We have created standard operating procedures for sampling in food industries, including also information about how to manipulate the samples and how to store them before processing for DNA extraction. These standard operating procedures are tailored to different production sectors. So we have sampling plans and information specific for many different food production sectors. And we will see then a video where this is a bit summarized, but I wanted to make some initial considerations about the main issues that should be taken into account during sampling. So first of all, it's very important that the personal sampling wears personal protective equipment, so do not influence with our microbiome, the microbiome of the sample we want to take. It's also very important to sample following the food chain production flow in each of the factories that we want to analyze. And when dealing specifically with food processing environments, it's very important to take into account what kind of tools are we going to use for sampling. What is the way we are going to sample? So what is the surface? So what are the individual units we want to sample? And also how to organize all that sampling in the industry. So in our case, we made some recommendations that you will see later on in the video. So first of all, we decided to use sponge stick swabs made of polyurethane, because these ones allow to recover much better the microorganisms in the environments, and also because they don't contaminate the sample with foreign DNA. So they are synthetic and they are totally DNA free. So these were the reasons why we decided to select this kind of sponge stick swabs that you can see now here in the slide. So we normally sample very wide surfaces, and we recommend sampling surfaces of around 1 square meter whenever that is possible. So in very open spaces like floors, walls, etc. And in other types of samples, or other types of environments where this is not possible, we recommend to sample individual units. So the whole drain, the whole knife, etc. Would be taken, would be swabbed and used as a sample. And we also recommend for each of the sample categories to pull several swabs. So let's say we are interested in knowing the microbiome of the knives. So we will be sampling 5 different knives, 5 individual knives, each one with one of the swabs, and then we will be pulling the 5 swabs in a single sampling bag. Why is that? Because as normally in food processing environments, we are thinking about very clean environments with very low microbial biomasses. We want to maximize the amount of microorganisms that we can recover and therefore to be able to get a sufficient amount of DNA for the microbiome analysis. Then all these pools of 5 swabs will be put together in a cooler and some device for transport and also for maintaining a low temperature the samples for transport to the laboratory. In our project, in our validation activities, we perform one single sample visit per industry. We did that at the beginning of the manufacturing day, when the surfaces were totally clean from the shift of the previous day. We were sampling different rooms within each of the factories. So, for instance, for a cheese maker, we will be visiting the processing room, but also the ripening room and the packaging room, if available, etc., etc. And for each of the rooms, we were sampling environments, mainly food contact, but also non-food contact environments. So food contact would be the knife, table, the vat, or the tank, and non-food contact environments would be the floors or the drains, but also the raw materials, in this case, the milk, the intermediate products and the end products, so in this case, the cheeses at the beginning of ripening and at the end of ripening, and the operators. So these are the examples of the sampling plans for a couple of products, for a couple of sectors. Let's say this is a sampling plan for a company producing fermented sausages, where we can see the type of samples we took from the processing room, the cold room, the ripening room, the smoking room, and the packing room. For sure, each factory needs, each production sector needs a specifically tailored sampling plan because some of the rooms only exist in particular sectors. And also some of the equipment is very specific for particular sectors. Here you can see also an example of another sampling plan, in this case for a cheese production facility. Okay, once we had all the samples and we transported them to the laboratory, so let's talk about the next step of the protocol, that is the manipulation of the samples in the laboratory before DNA extraction. So here, what we aim to do is to recover all the microbial population that we got in our swabbing activities in the different sampling bags. So for that, we will be using slightly different approaches for different types of samples. So for processing environments, we will have these bags with a pools of 5 swabs. And what we are going to do is to add a small volume of a buffer, phosphate buffer, saline, which is added to the sampling bag. This is a small volume, like 10 milliliters. Then we will be homogenizing the sample, normally using a stomacher at maximum speed for 2 minutes. And then we will be recovering the liquid, where we know that we will have all the microbial contaminants in the swabs. So after this homogenization, we are able to get all the microbial contaminants in our samples. We will be collecting that into a sampling tube, and we will be centrifuging the sampling tube so that we can get a pellet with all the microbial cells that either we use directly for DNA extraction, or either we store at low temperatures, at the freezing temperatures, for long-term conservation of the sample before DNA extraction. All this sample manipulation and recovery of the cellular pellet should be done quite fast after the sampling, ideally after a few hours, but for sure less than 24 hours from sampling. When we are dealing with other types of samples, let's say the food samples or the liquid or semi-liquid samples, as could be the milk, or the food samples as could be the cheese, we should use slightly different sample manipulation approaches. So for liquid samples and semi-liquid, semi-solid samples, it could be the milk, the water, meat, butter, etc., we are going to use sterile plastic tubes for collection of the sample in the facility. And what we are going to use is to follow a centrifugation procedure, followed by several serial washes with the phosphate buffer. And then after several rounds of centrifugation, we will be keeping the pellets also for storage at long term and the freezing or for direct DNA extraction. For the solid samples, foods such as cheese, sausages, etc., we will be using sampling bags in the facility, sterile sampling bags. We will be collecting samples of around 10 grams, and then we will be diluting 110 dilution with 90 milliliters of the phosphate buffer. This will be homogenized in the stomacher, maximum speed for a few minutes. And then the liquid will be recovered, and after centrifugation and several serial washings, similar to what we see here for the liquid samples with the phosphate buffer, we will be keeping also the pellets and storing them at freezing temperatures. Okay, so let's see now a video where this is summarized in a few minutes, where probably some of the doubts you may have will be clarified. Okay, so the first and most important thing to start with is the operator doing the sampling should be equipped with personal protective equipment. So the aim is not to influence ourselves the microbiome of the processing environments or of the samples, the food samples or the liquids that we want to analyze. So for that, we are going to use caps, shoes, protective shoes, and also gloves. It's very important also to have very well organized all the material we're going to use during sampling. So to have at hand all the marked, if possible, all the different swab bags, the tubes, the sampling bags for the foods, etc., so that we can do it all the sampling as fast as possible, disturbing as less as possible also the work of the company. So here we can see the polyurethane sponge swabs with the stick that we are going to use for sampling. You can see that the stick is very easily removed after sampling, and we're keeping only the sponge in the bag. And here you can see that 5 different swabs are being pulled from the same sample category. So it's a category of non-food contact surfaces. You have seen the sampling of drains, floors, and now you are seeing the sampling of a wall of the company. You can see that around 1 square meter is sampled using the swab in different directions. As I mentioned, the pulling of the 5 swabs is done mainly to maximize the recovery of microorganisms and therefore to ensure that a sufficient quantity of DNA is going to be obtained. Once the sample is taken, the pulls of different swabs are kept under the cooler at low temperatures. You can see that between samples it's recommended to change the gloves so that we don't influence from one sample to another. Here you can see that food samples are also collected in sample bags, in this case cheese, also tubes with liquid matrices like the milk. You can see now also the sampling of an operator, where either directly the hands or the personal protective equipment will be sampled. to see the influence of the operator in the final microbiome. And also it's possible to take air samples to know they can be used either as negative control or actually to know the impact of the air of the company in the microbiome of the product. So once we have all ready, we will be coming back to the lab for doing the sample manipulation steps. We will be working, if possible, in a cabinet, flow cabinet, and laminar flow. You can see that first for the environmental swabs, we will be adding the small volume of the buffer. We will be homogenizing this pool of swabs with the buffer for a couple of minutes on maximum speed. And then we will be recovering the liquid from the bags. Normally for that we need to squeeze a bit the sponge swabs because they retain some liquid. We can do it from the outside of the bag while we pipette out the liquid. You can see that this liquid is already with some turbidity. That means that it contains already a number of cells, mostly microbial cells. It can contain also other contaminants from the floors or from whatever the sample it is. We will be centrifuging those samples. And here we see already the cell pellet that is formed for sedimentation of the cells. We will be disposing of the liquid and keeping the pellet either for direct DNA extraction from the pellet or for storage in the freezer. For the food samples, we will be adding a higher amount of buffer. So it's a 110 dilution, 10 grams of product in 90 milliliters of buffer. In these sterile sampling bags that we use for collecting the sample. Then we will be homogenizing the stomacher chain. Finally, we are recovering the liquid, and we will be performing a series of centrifugations and washings with buffer of the pellet, keeping the final pellet after several serial washings for storage in the freezer. So all the samples, after being pre-processed, are stored in the freezer. We recommend storage at -80 if available, but if not, it can be done at -20. Of course, if possible, it's also recommended to do direct DNA extraction. That is quite frequent to keep the samples for long-term storage before analysis. Okay, so once we have seen this small video demonstrating the main points related to sampling and sample manipulation, let's go to the next steps of the protocol, and we will continue with the DNA extraction. So in this case, when we started master, we knew that we had some important challenges analyzing this type of samples, mainly the food processing environments. The first ones the first main challenge is that the samples from food processing environments are expected to contain very low microbial biomass, so very low abundance of microorganisms, and this difficults the sequencing and the obtention of sufficient DNA for sequencing and for getting meaningful data. But also we had another extra challenge, that is that also these food processing environments can be influenced by a series of inorganic and organic contaminants, including residues of sanitizers, food matrices, etc. And this could also interfere with the follow-up steps of the process, DNA extraction, or even the preparation of the library for sequencing, etc., etc. So this was a challenge. It was also a challenge, the fact that there are no available extraction kits in the market for this particular type of samples. So there are a lot of extraction kits in the market for DNA extraction, for genome sequencing, or for metagenome sequencing. But these have been optimized for other types of samples, mainly for human samples, human feces, or even for soils, for water, for other types of samples. So we wanted to test and to see whether we can get a protocol specifically tailored to this type of samples to get very high DNA yields for these very difficult samples. No? And we were lucky enough that one of the partners of Master is Qiajin, that is, as you may know, a leader in the market of DNA extraction protocols. And Qiajin have worked with us in the last few years very intensely to try to develop this protocol that would be that could have a high impact for our activities in the project, but also for food industries and for analytic laboratories in the future. So Qiajin developed a series of protocols, slightly different between them. So starting from some of their kits, and for combinations of components of their kits, and with a slight modifications, and arrived to the proposal of 5 different possibilities. They are pretty much a very similar approach, but with very slight differences between the 4 of them. And these proposals of possible extraction protocols, extraction kits to use, were tested by 3 of us, by the University of León, by the University of Naples in Italy, and by Chagas Quinaela. And we tested them, collecting a lot of samples from food processing environments, actually from food companies, from pilot plants as well. And we follow the different procedures, the different protocols proposed by Qiajin, and measure the yield of DNA that we were obtaining, so the nanograms of DNA per extraction. And as you can see, we saw important differences between the different proposals, and we decided to select the one performing better. That is this one that you can see here, the 3rd one. And then this was the method that was tested and used in all the validations that I mentioned before. So in those more than 100 companies, more than 2,000 samples. So the selected DNA extraction protocol has as basis one kit that is a commercial kit of Qiajin, that is the DNA Power Soil Pro kit, but with some important modifications to increase the DNA yields. So the chemistry is mainly the one of this kit that is commercial, but there are some relevant modifications. Well, first of all, to let you know, almost every extraction kit in the market has some basic steps that are quite similar between them. So normally every extraction of DNA starts with the lysis of the cells. Normally this happens either mechanically or using some chemical reagents. In this case, we are using a combination of mechanical and chemical lysis. So the main point here is to break the cell so that all the intracellular components are released. And then the next steps of any extraction protocol involves normally binding the DNA to a physical membrane that is coming in columns that are present in the kits, and washing and eliminating the rest of macromolecules and other substances that can be there in the sample. So eliminate the proteins, the RNA, or any other substance. the rest of the membrane cells, etc., and keep only the DNA fixed and bind to the membranes. Then in the last steps of the protocol, after several washings and purifications, what is normally done is to elute the DNA, so release the DNA from this binding to the membranes and recover it in a small volume where the DNA would be highly concentrated. So in our case, we are using, as I mentioned before, a combination of mechanical and chemical lysis. And then we are using some columns for DNA binding that are not the ones coming from this kit. There's another set of columns that are also from Kiakin and that are specifically tailored to get high DNA yields from low biomass samples. So because they allow eluting the DNA in very small volumes, and this has this advantage that you can increase the DNA concentration. Then there are also some adjustments and some modifications from the original instructions and steps of the kit. That is to add some changes in the washing steps, adding isopropanol in some of the steps of the washing steps. And this improves also the nucleic acid yield as shown by Kiakin. So this was the protocol that we applied in all the industries and in all those samples that I mentioned before. Okay, so once we have the DNA sample, we can either sequence it directly, or we can also keep that DNA sample at -80 degrees, also under freezing, for long-term storage. And what we will have in that DNA sample is a mixture of the genomic material of many different microorganisms. So we are recovering microbial biomass for maybe thousands of microorganisms in that sample. It will include the molds, yeasts, bacteria, etc., etc. And we will have all their genomes. Normally, we will have them fragmented because in the DNA extraction process, normally the DNA is fragmented to some extent. And we will have all this mixture of fragments of DNA coming from different cells. And what we will do is normally to also, using some kits, process that DNA for sequencing directly in a platform. Normally this is done by companies, external companies, sequencing services. So we normally just send them the DNA and they will prepare the library for sequencing. This is quite a standardized process where they add some small fragments of DNA, some adapters, and they do some amplifications, etc. It's a complex process, but quite standardized. And then the DNA is sequenced. Different platforms can be used for sequencing. In our project, we use the Illumina NovaSeq platform. Illumina is one of the leaders in the market of DNA sequencing, and this platform allows to get very high sequencing depths. Normally with Illumina, you will get very short sequences, so there's small fragments of DNA sequenced. But there are other platforms in the market. So this protocol, and we have validated it for sequencing with Illumina NovaSeq, but it could be for sure used with other platforms. This could be PacBio, Oxford Nanopore Technologies, etc., etc. Each of the platforms has some advantages, some limitations. For instance, with Oxford Nanopore Technologies, probably you do even in situ sequencing in the industries. You could get larger fragments of DNA, and that could have some advantages. But I mean, this is quite interesting to discuss, but it would need much more time. So, but in any case, we decided to use this Illumina NovaSeq platform, and the validation has been done with this platform. And basically, what we will get from the company doing the sequencing is a bunch of files where the sequences, the reads coming from each of the samples will be available. And then we will have to, bioinformatically, computationally, analyze those files and those reads that we obtain. And this is the last step of the protocol. So actually, this sequencing reads that we obtain from each of the samples contains the genetic code of each of the microorganisms. And this genetic code is very specific for each organism. So it's like a fingerprint. So if we can get this genetic code, we can say that, okay, this is coming specifically from this species or from this genus because it's very typical, and we can match our sequences with databases, and we can know exactly what kind of microorganisms we have in the sample. So we normally, in this bioinformatic analysis, previously we will be doing some checks of the quality of the reads. We will be removing the reads that are of bad quality. We will be removing parts of the reads that we have added during the library preparation and that are not interesting anymore, etc., etc. And then we can analyze all those sequencing reads in many different ways, and we can get a lot of very useful information. So first, as I mentioned, as this is a fingerprint, we can say, okay, who is in the sample? What kind of species, what type of microbes, what type of species, genomes, etc. we have in the genera we have in the sample. And if we analyze and compare particular samples in the industry, the foods, with particular environmental samples in the industry, we can even make hypotheses of what are the sources that are having a higher influence in the microbiome of the product, or what could be the source of a particular microorganism that is in the final product, etc., etc. But we don't only are able to say who is in the sample. We can even do more detailed analysis on the functionality of that community. So what kind of genes are in that sample? So not only who is there, but what are they able to do? No, are in the sample genes related to antimicrobial resistance to disinfectants, to virulence, to production of biogenic amines, to production of particular flavors, or flavor compounds, etc., etc. This can have very important. This can be very important information from a safety point of view, but also from a quality point of view. So even it is possible to assemble the source reads that we obtain. The assembly process is a complex analysis that is done computationally, where normally you can put together source reads like if you were doing a puzzle and you are getting longer and longer fragments. If you have a sequence within enough depth, you could even be able to reconstruct genomes from the sample. So coming from a mixture of DNA from different strains, different bacteria, different yeast, molds, you could even be able to reconstruct genomes of a few of the strains that you have in the sample. And this, of course, opens very important perspectives to see if some of these particular strains have features that would be of relevance from the point of view of the safety and the quality of the food. In fact, in our case, from the thousands of samples we analyzed, we were able to sequence more than 90% of them with very good sequencing results, with very high DNA yields, especially for these processing environment samples that we were a bit afraid that they could be very challenging. So these are the statistics on the total DNA yields for those food processing environments. So around 50 nanograms per microliter of mean, but with a lot of samples having a sufficient amount of DNA for Illumina sequencing. We obtained around 60 million reads per sample, and we were able to reconstruct, as an average, around 12 genomes in each of the samples, having some samples with around 60 genomes reconstructed. So this is significantly improving what was available in the literature from previous expertise doing sequencing of food processing environments. So just to summarize and to wrap up, the deep investigation of the microbiome of food production and processing environments through metagenomics has a huge potential and huge potential benefits for food safety monitoring programs, but also for food quality checking. There were several important technical challenges that had difficulted the application of metagenomics with this aim, the most relevant being the difficulty to recover a sufficient amount of DNA from this type of samples. And what we have done in Master is to develop an improved complete workflow with optimized sampling, but with a specifically developed and tailored DNA purification methodology that has been validated to obtain high-quality sequencing results from samples taken from food business operators for microbiome mapping activities. And the application of this protocol in a very large number of food processing plants across Europe allowed to get very high DNA yields, over 10 nanograms in more than 98% of the samples that we analyzed from food processing environments, which is sufficient and to to do a good quality Illumina sequencing for the microbiome analysis. Yeah, and that's it. So I hope you enjoy the webinar and that you find it useful for your professional activity. Thank you very much.